Okay guys, so this lecture is going to be a little bit different than our normal lecture. Usually you guys get a screen capture for these, but for this particular chapter, chapter 10, I'm going to be doing this by hand as you guys can see, so I'm going to be writing along with you guys. I don't really like the way that this lecture is laid out in the sense of teaching it over a screen capture on my computer, because it's very hard to draw out Punnett squares that way. So let's go ahead and get started. Chapter 10 is going to cover kind of genetics and how to determine what offspring are going to look like between a pairing of two parents. So if you breed animals or something like that, and you have a red dog and a brown dog and you breed them together, what colors will you get? Or if you have a birds that have a certain color that you like for feathers, how often are you going to get that particular feather color and things like that from genetics? How likely are your children to have certain hair colors and things like that? So that's what the chapters are going to talk about. Let's go ahead and start that. So the study of genetics, which we talked about a little bit, is going to explain how organisms look and how they function and how they're put together. So you guys can see there that cat with her kittens, the mother cat. Her offspring are still cats. They're the same species as her, but they look different than her. So genetically speaking, they are the same species. They have the same genes that make them a cat. But the genes, inside that make up the color of their hair, the color of their eyes, the color of their skin, and things like that, doesn't really matter for their survivability as a species, so those things can be different. You're going to be a cat regardless of what color fur you have, of things like that. So that's kind of how genetics works. Genetics allows things to be one way, as in the sense of being a cat, you are a cat species, but it also can shake up how you look as a cat species. You can be a different color cat, the longer hair, shorter hair, and things like that. So if you look at parents and you know how they breed, if you made a white cat to a white cat, they're going to have white babies 100% of the time, or a black cat to a white cat and you have spotted babies 100% of the time. You can figure out how these things work. You can figure out the ratios of what the chances of getting white babies or spotted babies or brown babies or black babies or whatever from breeding cats, what the likelihood of those particular patterns of fur coming out as the babies coming out from the breeding patterns, the matings, what those chances are going to be. So if you're breeding for a particular target, say you want black and white spotted kittens, you breed a white cat and a black cat together, and that gives you black and white spotted kittens 100% of the time. If you know that, you can use that to predict what would happen if you breed different color cats, or if you want to breed a more spotted cat with more spots, you might breed a spotted cat to a solid black or a solid white cat, which would bring in more white spots and more black spots and things like that. So you can use this knowledge of genetics to talk about and figure out how offspring are going to look between pairings of two parents. So most of the genes in your body, like I mentioned just a second ago, don't really have anything to do with how you look. They just make you you. They make up your hair. They make up they make up the structure of your hair, I should say. The fact that you have hair. They make up the fact that you have bones. The fact that you look the certain way that you do in the sense of you're a human. But those genes just make proteins. They make you function. They make the structure of you. But the differences in between those little subtleties of genes are what makes different color hair proteins, different color eye proteins, different color skin proteins, fur, hair, things like that, for animals across the planet, and same with us and humans. So if you guys recall back, DNA is wound really tightly into chromosomes. And this is how we can talk about genetics. So you guys remember, we have one chromosome here. You can see our sister chromatid. You can see one on either side. It's chromosome 1 from mother, copy 1, and chromosome 1 from mother, copy 2. Chromosome 1 from dad, copy 1. Chromosome 1 from dad, copy 2. So that's how genes work. Genes are going to be found on those chromosomes, and you can see that here. These little they're called locus, and that's the location of a gene on the chromosome itself. So every single chromosome, the locuses are set. So chromosome 16 is going to contain, I don't know this for sure, I'm just kind of making this up as I go along in the sense of what's on this chromosome. But chromosome 16 might contain genes that, or contain eye color and hair color and things like that. So you might have chromosome 16 that contains the eye color blue and the hair color orange or dark red kind of thing. Whereas someone else next to you on their chromosome 16 has the exact same thing for eyes. They have the same gene for eyes. It's just a different version of that same gene. And instead of having the gene for orange colored hair, they have a gene for, let's say, black colored hair. Hair was my little horrible drawing for hair there. So that's how that would work out. And that's how these things work. So chromosomes have the genes attached to them. The location of that gene on the chromosome itself is called the locus. And the locus doesn't change from chromosomes. What does change, or can change, however, is the gene itself that's at that location. So the locations are set in stone. Chromosome 16 is going to contain the gene that controls eye color. So it's going to be there regardless. What version of that eye color gene that you have is going to be different. So when you reproduce, your chromosomes are going to be split up via the process of meiosis. So you start out with a cell that has chromosome 1 from mother and chromosome 1 copies from mother. Now, if you guys recall, that chromosome is going to be split via the process of meiosis into this kind of thing, and then it's going to be split again into 4 cells that contain 1 copy of each chromosome. So then what's going to happen is in father, the exact same thing is going to happen. He's going to have his copy of chromosome 16 from himself, the mother and his father. It's going to be replicated and split via meiosis into that, which is then going to be split again into 4 sperm cells, which have 1 copy of that chromosome each. So that's the concept of diploid cells, making haploid with 2 copies into diploid down here. Or sorry, diploid with 2 copies into haploid with single cells down here. So how do we get a human? Well, you unite sperm. So the little sperm cell is going to have 1 copy of chromosome 16 with the egg cell from the mother, which is going to have 1 copy of chromosome 16 in it. And when you unite those 2 egg and sperm cells together, you're going to get an embryo that contains 1 copy of chromosome 16 from mom and 1 copy of chromosome 16 from dad. So that's how genetics works. That's how you shake up genetics. That's why you look like a blend of your mother and your father. Now, whichever way this shakes out depends on how you look. It depends on how chromosomes cross over, and multiple other things if you recall back onto our previous lecture sections about crossing over and things like that. So this is how this one works. Depending on, you guys recall that chromosome 16, maybe this red spot over here could swap with a little red spot or the little blue spot over here, and you end up with different unique combinations of chromosomes and things like that through crossing over. So very interesting processes with genetics. So this is the process of taking a diploid cell, an egg cell, or sperm-producing cell, egg-producing cell, sperm-producing cell, and then taking those diploid cells, producing 4 haploid eggs, or 3, technically 1 haploid egg, 3 polar bodies, 4 sperm cells, which then unite that are haploid to form a diploid zygote back to the start. So that's a quick little rundown of how genetics works in the sense of how reproduction works. You get 1 copy of your chromosome from your father, and then another copy of your chromosome from your mother. So humans have 23 chromosomes. We have 22 autosomes and 1 set of sex chromosomes. So you get 1 set of each 22 autosomes from your mother, including 1 X chromosome that comes from your, so this is sets. So 46 chromosomes in total. So you get 23 sets of chromosomes from your mother. So 22 autosomes are split in half. So you're going to get 11. You get 23, sorry, I split that in half. So you get, it's 46, so you get 23 from your mother, which is going to be 22 autosomes and 1 sex cell. And then you get from your father 22 of his autosomes and 1 sex chromosome. So from your mother, she can only give an X. From your father, he can give either an X or a Y, whatever that happens to be, or Y. So these 2 things are going to combine back. 22 plus 22 is 44, plus the 2 together from this X and X, or X and Y, gives you 2 more. So 44 plus 2 is 46, back to the original amount of chromosomes that humans have from the get-go. This is how this works. Really quick down and dirty breakdown of reproduction, of genetics, and things like that. So why does this matter? Why did I go over this stuff real fast about this one copy of chromosomes from your mother, one copy of chromosomes from your father kind of thing? So this is where the concept of genetics is going to come back into play here. So this guy right here, Gregory Mendel, very important guy. He figured out the ratios that organisms reproduce by. And this is to say, if you have black hair and you reproduce with another organism that has black hair, 100% of your offspring will have black hair. Or if you have black hair and you mate with someone who has white hair, all of your offspring will have gray hair. Instead of black and white, spotted hair and things like that. So he figured out how these ratios work, how blendings of colors work in animals and plants and things like that, how to select for certain traits and things like that. So let's talk about how he did this, and then let's talk about how it works. So Gregory Mendel, this was about 1830, 1840. This was quite a while before Charles Darwin, so evolution hadn't even been really solidified as a theory yet. So he didn't really understand what was going on here. Anyway, Gregory Mendel was a monk that lived in what is now present-day Austria. At the time, it was part of Germany, but now it's present-day Austria. Well, anyway, he was a monk, and he was tasked in the monastery that he lived in to run the monastic garden. So he was the gardener for all the monks and with their food. So I imagine it's kind of boring being a monk. There's not a lot going on at the monastery every single day, and you kind of have to be stuck there. So he liked to experiment in his garden. He was kind of a scientist at heart. He liked to do little experiments and things. And while he was playing around with the pea plants that he was growing in his garden, he noticed some things. And what he noticed were that pea plants came in two different colors. When they grew, the flowers, when the plant flowered, would be either white or purple. And that was kind of interesting to him. So the first year he planted them, all of his plants came up purple. And it was kind of cool. So the second year he planted them, he didn't really think anything about it. It's the second year. And they came back mix of purple and white. That's kind of interesting. Where did the white come from? There were no white plants in there earlier. That's kind of strange. And the next year he bred them, he got pink, white, pink, and purple. That's kind of even weirder. So that's kind of strange. Where did that pink come from? There was no pink or in the original plants, the purple. There was no pink in the second year plants, the white. So how does this work? So what he was doing is he was collecting the plants at the end of the first year, collecting the seeds, and then replanting them the next year to restart his harvest. So he was using the same batch of seeds every single year that had been grown from the previous year's harvest. So he noticed all of these very interesting things. And what he also noticed were that sometimes the plants grew tall, sometimes they were really short, sometimes the peas inside of the pods were yellow, sometimes the peas were green, sometimes they were wrinkly, sometimes they were smooth. And he noticed all of these different traits in the different types of pea plants, or in the different pea plants that he planted. It was all the same species of peas, but they all looked different. So he noticed all of these different traits, and he set out to figure out how this thing worked, how these traits, these looks, these pink flowers, how he could get pink, how he could get just purple, how he could get just white, get just yellow, get just green seeds and things like that. He set out to figure out how inheritance from one generation worked to the other, how the chromosomes were split, how the genetics shake up, and things like that. Now, you have to remember, he didn't know what genetics was. He didn't know what DNA was. He didn't know what chromosomes were. He didn't even know what genes were. So to him, he was just doing this based on pure observational science, what he could see, which is really cool. And he was able to figure this out very accurately using solely methods that he could see, and not actually going down to a genetic level to get far into this to figure out the results. He actually just did this based on pure observational science, which is really cool. So the first thing that we need to talk about is something called a true breeding plant. So what Gregory Mendel did is he figured out that if he bred a purple plant to a purple plant every single time the offspring would be purple. So what he would do is he would go out and he would take a pea plant that was purple, and he would find another purple pea plant, and he would take the pollen pods, he would cut them with a pair of scissors, and then he would purposefully pollinate the plant so he could control who it mated with, who it bred with, and not just random mating. So he was controlling who it mated with, which is how he knew what the genetic ratios would eventually be like and what the parents were. So anyway, you take a purple plant, a purple flower plant, and you breed it to another purple flower plant. Every single time you do this, all of the offspring, every single time, no matter how many times you do this, will be purple. If that's the case, that plant is considered a true breeding plant. It mates with itself, or you mate with the another version of that same plant, and you're going to get the same plant, the same color, identical every single time. So you can get a tall plant with green seeds and purple flowers, or a tall plant with yellow seeds and white flowers every single time. So as long as that plant produces an exact copy of itself every single time that they're born, that the offspring are born, that is going to be considered a true breeding plant. So you need to know what is true breeding and what is not to be able to figure out what the genetic ratios are. So if you know every single time that gene's going to, or that cat is going to have brown babies every single time it mates with another brown cat, you know you've got a true breeding individual. Now you take those two true breeding individuals and then you start mating them together. And then you can figure out the differences between the types of genetic makeup, which is really interesting. So how this works. We talked about just a second ago how you have one copy of chromosome from your mother and another copy of that chromosome from your father. Now, sometimes one version of that chromosome, or both, are considered dominant, meaning that the gene that's found on that chromosome, whatever it happens to be, is going to be the gene that whatever is over here, if you have a gene for blue eyes over here on your mother's side, and a gene for, let's say, green eyes on your father's side, that blue-eye gene is going to come over here and beat up that green-eye gene, and you will have blue eyes no matter what. If that is the case, this gene is considered dominant. It will dominate every single other gene around it. It will overpower the other gene that's found on the matching chromosome, the sister chromatid, every single time, no matter what it is. So that's how that works. If you have one copy of a dominant gene on your mother's chromosome or your father's chromosome, as long as there's one copy of that chromosome somewhere inside your genetic code, you will express whatever that genetic makeup wants you to have. So if that one dominant gene says you're going to have blue eyes or brown eyes or whatever, you will have those color eyes no matter what the other gene says. So that's how that works. So the other hand of that is something called a recessive gene. So this is how this one works. If you have your mother's chromosome here and your father's chromosome here, and on there the colors, genes for eye color, your mother has blue eyes again, your father has green eyes. In this case, what's going to happen, we'll do the exact same scenario. You're going to have blue eyes because this gene is dominant and this gene is recessive. And that means that it's kind of the wimpy gene. It's going to not have enough power to make itself shown. It's not going to say, oh, you're going to have green eyes. It's going to say, oh, okay, Mr. Body, you can have blue eyes. That's perfectly fine with me. I'm not going to express at all. I will not cause any proteins to be made that will cause green eyes to show up. So I'm going to just essentially be dominated by that dominant gene and turn off. So that's how this works. If you have one dominant gene, no matter what the second version of your gene is on the chromosome, remember you have two chromosomes, one from mother, one from father, and you get one copy of each gene from your mother and your father. So one gene from your eye color from your mom, one gene from your dad. So if mom's overrides dad's or dad overrides mom's, it's a dominant. You have to have one version of a dominant gene to override the second one. Whatever the second one is does not matter as long as you have one copy that's dominant. Now, for recessive genes, how do we get an individual with green eyes? Well, green is a recessive gene. It has to be expressed twice. It's a little wimpy gene. It's not very powerful. So to get an organism with green eyes, you have to have 2 recessive genes show up in the genetic code. So one dominant gene will always overpower one recessive gene. So it doesn't matter if you have one recessive gene, you have to have 2 of them to be able to have green eyes in our little scenario here. And this is how this works. So let's go ahead and talk very quickly about how we label this stuff. So let's stick with our example of eye color. So let's stick with green eyes and blue eyes. Actually, let's stick with, let's just switch this to black eyes and blue eyes, or brown eyes and blue eyes. Let me get a brown colored pencil real fast. All right, so let's switch to brown eyes and blue eyes. And this is a real world scenario here where brown, let's go brown, in dominant letters, brown is a dominant gene, which means if you have the color, the gene in your genetic makeup, on your chromosome that contains the allele for eye color, if your chromosome says that you're going to have brown eyes, that is a dominant gene, which means that it will express itself no matter what the second chromosome from your father or your mother, whichever the other one happens to be, is. So as long as you have one version of that dominant gene, you have one brown eye gene, whatever the second gene in your genetic makeup is will not matter. So a dominant gene, in this case scenario, we're going to go with eye colors. We're going to go with brown, is given the letter B, an uppercase B, meaning dominant. Okay, so we know it's uppercase, we know it's dominant because of the uppercase concept. So, recessive genes. In this case, this is going to be kind of odd. Now, our eye color is brown. Now, in this case, it's anything other than brown, and this is kind of unfortunate how this works. So, if our dominant gene is brown, our recessive gene, anything that's not brown, is going to be a lowercase b. Now, in this case, I've told you what the eye colors are, brown and blue. So how I would present this on the exam is you have an organism that is a brown-eyed, true breeding individual. Okay, so if you have a brown-eyed, true breeding individual, you know that every single time that that organism has a baby, that they're going to have brown eyes. So what you would do is you would look at this and you would go, oh, how does this work? Well, first off, this is a true breeding individual. For a true breeding individual, we know from our earlier experiment, we have to have 2 dominant genes, or 2 of the exact same version of that gene, not dominant, 2 of the exact same versions of that gene to get an identical version of the offspring. So in this case, our dominant gene is brown. So if it's a true breeding individual, that means brown is dominant, and every single time it's going to have a baby, we're going to have brown-eyed babies. The first gene is going to be a dominant B for brown. That's good. It's going to give us the brown. And the second one's also going to be a dominant brown. We know that because it's a true breeding individual, which means there's no shakeup. This B is going to go to the egg. This B is going to go to the sperm. To one egg. This B is going to go to the other egg. And they're split up, duplicated, split to 4. So every single cell, when it reproduces, gets the exact same genes inside of it. So this is a true breeding individual. Every time this organism replicates, you're going to have 4 eggs or 4 sperm. Remember, this is going to be replicated duplicated, I should say, before meiosis takes place into 4 cells. Those 4 cells are going to split into this, one big B in each of the cell. So how this works in the other side, let's say you have a father that has blue eyes. So in this case, blue is anything other than brown, which is our recessive gene. So your father is going to be blue. He's a true breeding, blue-eyed individual. We know this because blue is a recessive trait. I told you that at the beginning. And to have blue eyes, we know that since it's a recessive trait, you have to have two versions. One from your mother, one from your father, two versions of each one, or one version of each from your mother, one from your father, to have those blue eyes. Because if you have anything other than that, any dominant gene is going to overpower that recessive gene. So if you have a recessive gene, you're not going to have blue eyes. So to have blue eyes, you have to have both those recessive genes. So this male, when he's going to produce sperm, we know it's going to split up into 4 sperm cells, which are going to produce this. So actually, I'm just going to put a circle around that because that worked pretty well. Okay, so 4 sperm cells with little bees inside of them. When these two parents get together and have a child, every single time that these sperm and eggs are going to unite, you are going to get this in their offspring, or baby with one dominant, one recessive gene. So in this case, our baby's going to have brown eyes as well. Okay, and we know that because he has a dominant gene that's brown and a recessive gene for blue. The dominant gene is brown. It's going to overpower that recessive gene here. So no matter what this gene says, you're going to have blue eyes. It's a weak gene. It cannot express itself. So in this case, this baby's going to be born with brown eyes because that dominant gene is going to overpower it. So a little quick breakdown of how this works. So if you are an organism that has 2 sets of dominant genes, 2 big B's from your mother, or one from your mother, one from your father, you are considered a homozygous individual. Okay, so homo means the same, zygo meaning zygote. So 2 of the same genes from your zygote. So same zygote, so 2 of the same gene. So in this case, our organism's going to be born, I switch colors, with brown eyes. Every time that this organism is going to produce an egg cell or a sperm cell, whatever it happens to be, the egg and sperm can only be brown. That's all that this organism can give to his egg and sperm. That's all he has to give. So that you can't give anything else. So the only option that this organism can give is brown eyes. So in this case, it's homozygous dominant. We know that because there's 2 big B's. Now let's talk about the other side of this. Let's talk about how we have blue eyes. Well, if you have blue eyes, you're going to have 2 recessive copies, and we talked about that earlier. In this case, it's also homozygous. But in this case, it's recessive. So when this organism is going to produce sperm or eggs, all 4 of those sperm or egg cells can only be little bees. That's all it has to give. So this organism is going to be true breeding, and this is also true breeding. That's all they have to give. Now, so in this case, what happens if you have one of each? If you have one recessive and one dominant gene. So if this organism got together and had a baby with this organism, what would happen? Well, this organism, our homozygous individual, can only give a big B. That's all you're going to get. This organism can only give a little b, and that's all you're going to get. So when you mix a big B and a little b together, you're going to get one of each, one big B and one little b. Now in this case, we know that this organism is going to have brown eyes, and we know that because we got that dominant gene in there. But how do we label this? This is called heterozygous. So hetero meaning different, zygote meaning zygote itself. So hetero, different zygote. So heterozygous, this organism is going to have brown eyes. You don't have to put heterozygous or recessive or heterozygous dominant here because there's one of each. It's different. You don't have to tell which one it is. It's that's what that word means, different. So it says it in itself. So we know this organism is heterozygous. So they're going to have brown eyes, but when they have a baby, they have a 50-50 chance of giving one sperm with a big B, or one chromosome with a big B, or in the other half of their cells, they could give one chromosome with a little b. So they have a 50-50 chance of having babies with the gene for brown eyes that they give in their sperm cells, or 50-50 chance of having little b, blue-eyed babies that are given in their sperm cells. So heterozygous individuals shake up the gene pool. Homozygous individuals are straight breeding. They remain the same. So given this information, you can look at the breeding rates and how likely you are to get particular traits that you're looking for in a population. So that's what I just went over there right there. Heterozygous dominant, homozygous, and homozygous recessive kind of thing. So let's talk about how we tell this apart. So this is mostly why I'm doing this. These Punnett squares are very difficult to draw on the PowerPoint slide. So a Punnett square. Punnett squares are quick and easy ways to predict the genetic outcome of a parent breeding cross. So what you would see on the exam, you'll need some scratch paper for your exam, so I'll put that on the exam to remind you. I'm going to give you a question that reads something very simple like this. A homozygous dominant father, first off, I would tell you what the trait is. So in this case, blue eye color, let's switch to hair. Let's make it red hair color. So let's use birds and be feathers. Actually, let's just do birds and feathers. So red feathers is dominant. So red feathers is dominant. If you have one dominant gene, you're going to have red feathers. And let's make green feathers recessive. Now, we write this as a lowercase r. I'm just going to use a green color marker here so we know what the representation is. So in this case, what we're going to do, we know that when this organism, so first off, I'm going to tell you, I told you what the traits are. So feather color, red is dominant, green is recessive. So this is how this is going to work on your exam. I'm going to tell you, you are going to breed together a homozygous dominant male and a homozygous dominant female. So you need to go through what that works through. So I'm going to write down here, homozygous dominant male and a homozygous dominant female. Okay, so our homozygous dominant male. Homozygous means two of the same, and in this case, dominant, the gene is red, red feather color. So he is homozygous, meaning he has two of the same genes, and that same gene happens to be dominant. So homozygous dominant red, he is RR. The male has the genetic makeup of RR, which means he has red feathers with two dominant genes inside of it. Our female, on the other hand, she's homozygous dominant as well. So in this case, she's also going to have a big R and a big R, two red R's, homozygous dominant female. So let's cross these and figure out what their offspring would look like. And what are the chances of getting a green offspring in this crossbreeding? So you draw a tic-tac-toe board, and that's called a Punnett square. And you can separate out the genes on this Punnett square and then do a cross. So let's put the male up at the top and the female on the side. You can switch these, it does not matter either way. So our male, he has when his chromosomes split up to produce sperm cells, they're going to separate. So one of these is going to go to one sperm cell and the other one's going to go to the other. Well, in this case, he only has one big R that he can give to one sperm cell. So we're going to put that in the first blank. And the second chromosome that he can give in his other sperm cell is also a big R. So we're going to put that in the second blank. Then we're going to move on to our female down here. Well, in her egg cell, she can either have one big R, that's the one option she can give in her chromosome, or the other option of her chromosome, which you remember when meiosis, they split up and you end up with 4 different copies of the cell. So she could have another big R, so R and R. Let's go ahead and cross these. So if these two organisms had a baby, you blend together, you just come to the middle and write down what happens to be together. So an R here and an R here, R, R, blend them together. R, and another R, R, and another R, and R, and R, and you get the idea. So every single time that these organisms breed, all of their offspring, let's go ahead and talk about this, are going to be homozygous dominant. You can just write this in there, HD, homozygous dominant, homozygous dominant, homozygous dominant. And what color are they going to be? Well, in this case, that's a homozygous dominant individual. That's going to be red. Also red, and also red. So we know using this Punnett square that if we bred together a homozygous dominant male bird with red feathers and a homozygous dominant female bird with red feathers, that every single time that they breed, their offspring will have red feathers and they will be homozygous dominant as well. So how do you report this results? Well, what you would report are two different things: the genotypic ratio and the phenotypic ratio. So the genotypic ratio looks at the actual number of genes that are found inside of the Punnett square cross and what the combinations of those genes are. The phenotypic ratio looks at how this organism looks on the surface, what its physical appearance is. So let's go ahead and talk about how we do this. I'll switch over to a blue marker so it's a little easier to see. So let's go ahead and talk about what these are. So let's do the genotypic ratio first. So let's go through and figure out what these guys are. We've already done this, actually. So this organism is homozygous dominant. This next organism is also homozygous dominant. This next one's also homozygous dominant and also homozygous dominant. So every single time that these organisms mate, there's a 1 in 4 chance, 1 in 4 chance, 1 in 4 chance, and 1 in 4 chance of producing one of these 4 pairings. You have a 25% chance of having either one of these offspring every single time they have a baby. So in this case, every single one of them happens to be the same. So we report our results as a genotypic ratio of 100% homozygous dominant. 100% homozygous dominant individuals. Every time they breed, they will always be homozygous dominant. So the other way that we're looking at, the other report, is the phenotypic ratio. What's the results of how these organisms look? So in this case, we've already done that as well. This is a homozygous dominant individual. Homozygous dominant, dominant, dominant. Well, our dominant trait is red feathers. So we know we've got a red bird, red bird, red bird, and red bird. So we would report our phenotypic ratio also as 100% red feathers. So that's a really quick example of breeding two homozygous dominant individuals together, which in this case is R and R and R and R. So red feathers individual bird with a red feathered individual bird. So let's go ahead and walk through a couple more examples of this real quick, and then we'll move on to a different idea behind these concepts. So let's draw another Punnett square. So in this case, we're going to shake it up a little bit. Let's breed a let's keep with the same traits, R and R, just for consistency. So red feathers and green feathers. So in this case, let's breed a homozygous dominant individual, so HD, with a homozygous recessive individual. So HD, HD, and HR. So let's go ahead and figure out what these are going to be. Homozygous dominant. Well, homozygous dominant is two of the same, and dominant in this case is R. And we're going to cross it with a homozygous recessive individual. Now that homozygous recessive individual is going to be different. Homozygous means the same, and two of the same recessive genes. So in this case, R and R. We have a green bird this time, something different. So we're going to breed a red bird with a green bird. So let's go ahead and separate out their genes on our Punnett square. Well, one red on the top and one red on this other square. One sperm cell could be red, and the other one that sperm cell could give in the other chromosome is also red. So that's all you get. And in this case, in our eggs, that's the same. We're going to split those up as well. Let's go ahead and finish our cross here. Big R, big R, big R, big R, little r, little r, little r, and little r. Okay, so that's the easy way to do this. A very quick cross between a homozygous dominant and a homozygous recessive individual. So let's report our genotypic ratio and our phenotypic ratio. So let's go ahead and talk and count about what these things look like. So first, let's do our genotypic ratio. Homozygous dominant, what are these? Let's go ahead and figure that out. Well, that's a dominant gene, and that second gene is different. Well, if we refer back, if you have two of the same genes, you're a homozygous individual. Then you have to determine if they're two of the dominant genes or two recessive genes. But if you have two different genes, then you're heterozygous, HZ. Oh, that one's the same, heterozygous, HZ, and then HZ. So 100% of these organisms, every single time that these organisms mate, have a 1 in 4 chance of having each one of these babies. All the babies are the same. So 100% of the time, these organisms are going to produce heterozygous individuals. And that's how that's reported. Now let's talk about what these guys look like. Well, homozygous dominant, recessive. Oh, nope, it's heterozygous. So in this case, we got a dominant gene and a recessive gene. We know that the dominant gene overpowers the recessive gene, so this organism is red. Same here, dominant overpowers recessive, red, and red. So in this case, they are 100% red. I should probably put 100% heterozygous on there so we know what that means. So with this cross between a homozygous dominant individual and a homozygous recessive individual, even though the genetics are different, we still end up with 100% homozygous individuals and 100% red individuals. So let's talk about how we get a green bird from this crossing. The only way to get a green bird back from these crossings is to breed one of these guys to one of these guys. So let's talk about this ratio here real quick. And this is an important ratio for Punnett squares. So let's draw our Punnett square here. We're going to have two heterozygous birds. So I'm going to draw up here, big R for red feathers and a little r for green. Now, in this case, we're going to have two heterozygous individuals. Hetero meaning different. So in this case, we know that our heterozygous individuals are going to have one big R for each and one little r for each. They're heterozygous. So we're going to cross our two heterozygous individuals, and then we're going to figure out what the ratio of their offspring are going to look like. So let's go ahead and split this up. We know that one of the sperm cells has a possibility to contain a big R, and one of them can contain a little r, and that's a little different. The other organism can donate an egg cell that contains either one big R or one little r, and that's how their egg cell works. So let's go ahead and continue this crossing. Big R, that's one organism. Big R, little r. Big R, little r. And little r, little r. Oh, that's a little different. So this is how you get a green bird. So in this case, let's go ahead and report our genotypic and phenotypic ratios. So let's figure out what these guys look like and what color they are and what their genes are. Now in this case, we have two big dominant genes here. That's a red bird. So this is going to be homozygous dominant, red. This guy, two of the different genes, one dominant, one recessive, is going to be heterozygous. Also heterozygous, HZ. And, oh, this one's different. We have two recessive genes here. Those two recessive genes are going to be homozygous. They're the same. So homozygous, but in this case, they're recessive genes. So homozygous recessive, HR. And what does he look like? These are red. They got that big dominant gene. Well, his dominant gene doesn't have one. He has two recessive genes. Well, what does that recessive gene code for? You have to have two of them to have that color. It codes for green. So this guy is homozygous recessive, and he's green. So let's go ahead and talk about this. Every single time these two organisms have an offspring, there's a 1 in 4 chance of having one of these. Every single time. So let's go ahead and report this. One homozygous dominant. Any more? Nope. So 1 HD. 2. How many is the other one? So that's the little asterisk is there, our little colon. 2 homozygous individuals, so 2 HZ, excuse me. 2 homozygous individuals. And then how many homozygous recessives? Just one. So 1 homozygous recessives. And that's how you would report this. So let's talk about our phenotypic ratio. What do these guys look like? Well, our phenotypic ratio, how many red ones are there? 1, 2, and 3. So 3 red. 2, how many other colors? Just 1. 1 green. So the ratio here is 1 to 2 to 1 and 3 to 1. And those ratios will always be the same as long as you have 2 heterozygous individuals being bred to one another. Heterozygous individuals always result in a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. So if you know you have recessive genes in your population, it's best to control them, or sometimes you want to enhance them. So this is how you know how to do that. You look for organisms that produce green or the recessive trait, whatever that so happens to be. And since it is a 1 in 4 chance of it actually being useful there, these guys are going to be what you're trying to breed together because you want to get this, or you can breed this together with another dual recessive R to get what you're looking for. So this individual here produces that second trait that you're looking for here. So that's heterozygous for heterozygous traits. So that's a very quick little rundown of homozygous dominant and homozygous dominant crossed together. Homozygous recessive crossed with homozygous dominant together, and then 2 heterozygote individuals crossed together. So the ratio will, for our heterozygotes, will always be 1 to 2 to 1 and 3 to 1. So that's a characteristic thing that's just easy to go ahead and memorize for a homozygous cross, or excuse me, heterozygous cross. 2 heterozygote individuals crossed together. So let's go ahead and move through our slides so real quick. What else we need to talk about? Okay, so sometimes genes can have more than one trait associated with them, because you guys, as you know, you don't have just hair. You have hair and eye color, or you have hair and skin color, hair and height, and things like that. You have more than one gene that all work together. So you can talk about traits that are carried together at the same time and look at individuals with more than one traits using slightly larger Punnett squares here. So this is going to be a little more fun, but it's the same kind of idea. So let's stick with our same traits here for birds. We have our red feathers, dominant. Green feathers are going to be recessive. And now what we're going to do is we're going to add in a second trait. Let's add in blue wings as dominant and orange wings, or little case blue, as recessive. And that's how this is going to work. Those are our 4 traits here. So let's go ahead and talk about what the parents are going to look like, and then we can go ahead and carry out our cross between these 2 parents. So let's talk about how we write this. So let's say we have a homozygous dominant red feathered bird with blue, that is dominant for blue wings. So let's go ahead and break that down. A red feather dominant bird, so 2 reds, 2 big R's, red feather dominant, homozygous dominant, individual. We write the first trait together with both of those 2 genes together. So if it was heterozygous, it'd be big R, little r kind of thing. Homozygous dominant bird with red feathers, with a homozygous dominant bird with blue wings, a homozygous dominant bird with red feathers and blue wings. So our second trait is blue wings. That's also going to be homozygous dominant. So RR, big B, big B. So homozygous dominant bird for red wings, or red feathers, with that's homozygous dominant for blue wings at the same time. You're going to be crossing that with the exact same thing. So let's go ahead and cross that with another homozygous dominant red bird with blue wings. That's homozygous dominant for those wings. So that's how that one works. So now let's go ahead and draw our super duper Punnett square. Okay, so here's our super duper Punnett square. Now in this case, we have to do the FOIL method. First, outer, inner, last. So let's go ahead and talk about our first individual. First, you're going to start here, and you're going to distribute it to the first letter. So big R, big B. So that's what's going to go in the very first block here. Big R and big B. Let's go ahead and foil first, outer. Okay, so the second square is going to contain the same, big R, big B. So foil first, outer, inner, and you're going to move to the red. Second one, inner, big R, little b, or big R, big B again. Last is the L in foil, first, outer, inner, last. So the same thing is going to go there, big R and big B. Okay. So if you look over here, we're going to do the same thing, big R to big B. Sorry. Outer or inner, so big R to big B, and last, big R to big B. Okay, and that's how you distribute out our Punnett square for these large Punnett squares, these two gene Punnett squares, looking at two different traits. So let's go ahead and fill in our Punnett squares. Let's go ahead and fill these in. So put the two R's together, you group the letters together. So two big R's and two big B's. Now I'm going to go ahead and do this real fast with my colored pencil, just because it's our color with my marker, just because it's easy. If you guys want to do this individually, you can do all the R's at once, do all the B's at once, kind of thing. Just keep them grouped together. Okay, I'm going to go ahead and add the B's. Okay, so that's a real quick way to break that one down. Let's go ahead and report our phenotypic and our genotypic ratios. Okay, so genotypic ratios and our phenotypic ratios. I'm not going to make you report the genotypic ratios. That would be horrible. You would have to go through and count all of these together. That would be not fun for these big guys. So for the genotypic ratios, we're not going to report that. That would be a lot of counting. But for the phenotypic ratios, they're easy to count. And we can do this very simply. So let's go ahead and count what these guys look like. Red bird with blue wings, RB, 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 RB. Okay, red bird, blue wings, and they're all red birds with blue wings. So our phenotypic ratio would be 100% red birds with blue wings. And that's a really quick little breakdown of crossing two homozygous dominant individuals on a large Punnett square. And it works the same if they're homozygous recessive as well. So let's go ahead and shake that up a little bit and cross a heterozygous individuals together for both traits. So let's go ahead and write up here, big R for red, little r for green, feather color, and then big B for blue feathers, and then on their wings, and then a little b for orange wing color. So in this case, let's cross two heterozygote individuals for both traits together. So in this case, we're going to have two heterozygotes for both traits. Trait 1 is going to be heterozygote, which is going to be our feather color, and trait 2, which is going to be the color of our wings, is also going to be heterozygous as well. So let's go ahead and write that out. So organism 1 we know is heterozygous, I told you. So heterozygous for trait 1 is going to be big R, little r. It's a red bird, but it carries that gene for green feathers. Our second bird is going to be, our second trait is going to be wing color, which in this case we know is heterozygous. He has a blue wing, but he also carries the gene for orange wings. And we're going to cross it with the exact same thing. Big R, big B, and little b. Okay, so two heterozygotes crossed together for both traits. Let's go ahead and draw our big Punnett square here again. Okay, let's divide out our chromosomes. So our first organism, so that's big R and a big B. So big R, big B, outer, or first outer, so that's the second. So first outer, oh, it's a little different this time. So big R and a little b, that's a little different. First outer, inner, move to our little r, and go to a big B. So that's a little slightly different. Little r, big B, last. So let's go here and go to that one. Little r and a little b. So that's a double recessive gene. That's interesting. So I'm going to go ahead and skip to this step simply because this one is the exact same as this one. So you can already work through it. So you can work through that one yourself if you'd like to. It's the same. So go ahead and write these down here as well. So R, B, R, B. Okay. Nope, I had that already start with, yeah, so R, and then B. Gotcha. Okay, cool. So now we're on the right page. I'm gonna highlight that B again so it's easy to see and it's front, blank, and center. Okay, cool. So that separates that out like it should be on our Punnett Square. So let's go ahead and cross these. So I'm gonna go ahead and cross these. You guys can cross these yourselves if you'd like to. I'm gonna make this easy all in one color at the same time. Okay, so I went ahead and put in all the big R's where they should be. Let's go ahead and put in all the little r's where they need to be. So it's a little r there. No little r's there, little r there, little r here, little r here, little r here, and a little r here. Oh, actually 2 little r's. Interesting. So one from there and one from there. Cool, that's kind of neat. So 2 little r's. Okay, and now what we can do is we can fill in our little r down here, 2 little r's and 2 little r's. Now we'll fill in our b's. So let's go ahead and fill in the big b's. So 2 big b's here, big B here, big B here, 2 big b's, 1 big B. Okay, so now let's fill in our little b's. So where do they go? Right here. And here's 1. There's 1. There's 2. 1, 1, 1, 1, 1, 2. Oh, I forgot to put in our little r there. 1 and 2. Okay, so that's how you cross 2 heterozygote individuals for both traits with another heterozygote with the same traits. Okay, so now let's go ahead and talk about our genotypic and phenotypic ratio. Once again, I'm not going to make you do the genotypic ratio. That would not be fun. You have to go through and figure out what's phenotypically or what's genotypically heterozygote for trait 1. That would just be way too much work. So let's go ahead and talk about what these guys look like. I like to give these guys squares, shapes, triangles, things like that, just because it's easy. So let's go ahead and label all the big squares that we see that look just like this. So RR, big B, big B. Let's give it a square. Okay, I don't see any more of those. Let's see if we've got any of these guys. 2 big R's, 1 big B, and a little b. Okay, let's give that a circle. 2 big R's, 1 big B, and a little b. 2 big R's, 1 big B, and a little b. Put my little r down there too. Okay, so 2 of those. How many big R to little r's? Big B to little b. So let's compare these together. So let's give this one a triangle. Okay, so Okay. So little r, big B, big B. Big R to little r, big B to big B. Okay. Oh, I'm thinking too much. Let's just go ahead and talk about what these guys look like. That's easier. So, red, wet feathers with, I'm doing genotypic ratio. Sorry, I should just be talking about phenotypic ratio. What they look like. I'm trying to think too much. Okay, so let's just talk about what they look like. That's the easier way. Red wings, red feathers with blue wings. Let's give him a square. Red feathers with blue wings. Let's give him a square as well. Red feathers, blue wings. Let's give him a square. Red feathers with blue wings. Let's give him a square. Red feathers with blue wings. Let's give him a square. We know that because he's got two. All he needs is one to have red. All he needs is one there to have blue wings. So red feathers with, now we got orange wings. Let's give him a triangle. Red feathers, blue wings. Let's give him a square. Red feathers with orange wings. Another triangle. On down here. Red feathers, blue wings. Another square. Red feathers, blue wings, another square. Oh, something different here. Green feathers with blue wings. That sounds kind of neat. Let's give him a circle. Okay. Red feathers with blue wings. Let's give him a circle. Red feathers with blue wings. Let's give him a square again. Red feathers with our orange wings. We already had that here, so he gets another triangle. Green feathers with blue wings. That's right, the same as above it. So he gets a circle. And our new one down here, which is totally different, is a green bird with orange wings. So let's give him a star. Okay, so now we got to tally up what all we've got. How many triangles, how many circles, how many squares. So if we did this correctly, we should have a ratio of 9 to 3 to 3 to 1. 9 that are red with blue wings, 3 that are red with orange wings, 3 that are green with blue wings, and 1 that is green with orange wings. So let's see if we did this correctly. So let's count up our red with blue wings, which we gave the square. How many squares? 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh, it looks good so far. We got 9 organisms that are red with blue wings. How many of our other organisms? Let's count up our next shape. So red with orange wings. This is a triangle. 1, 2, 3 triangles. Oh, we're looking good. So we have 3 organisms that are red with orange wings. Let's count up our next one. Our next shape would be the circle. How many birds do we have with green wings, with green feathers with blue wings? 1, 2, 3. We're looking really good again. So 3 birds with green feathers and blue wings. And let's see if we got one left over, or we got one shape left. That's a star, and that organism is green with orange wings. So this is the ratio for breeding together 2 heterozygote individuals with 2 traits. 9 to 3 to 3 to 1 for the phenotypic ratio of what they're going to look like. It will always be the same, 9 to 3 to 3 to 1 for the phenotypic ratio of a dihybrid cross. So this type of cross, the little simple, is called a monohybrid. And the big giant fancy one is called a dihybrid. Monohybrid, 1 gene equals the big A thing like that. Dihybrid is 2 genes. Like that. So that's essentially what that means, and that's what we're going for with that. So let's move on a little bit more. So let's go through all this stuff real fast. Here is an interesting little concept that can sometimes occur in genetics. And this explains why sometimes you get organisms that look a little different than both of their parents. So in this case, you can have something called incomplete dominance, codominance, or pleiotropy. So in incomplete dominance, you have so let's just stick with our bird example here, a bird that has red feathers and a bird that has, let's not go with green this time, let's say, let's say purple. Now in this case, purple is also a dominant gene. Different genes. So if a bird bred with purple feathers with a bird with green feathers, that bird would have purple. It would still overpower the green, since green is still recessive. I just added in an extra gene this time, purple. So in this case, let's breed a homozygous dominant red bird with a homozygous dominant purple bird. Homozygous dominant bird is R and R, and our homozygous dominant purple bird is P and P. Breed those together. Let's do our little monohybrid cross real quick. Got 2 P's on the side for one bird, one from each, and 2 R's on the top, one from each of the sperm and egg. Do our cross together. Okay, genotypic ratio and phenotypic ratio. Well, let's go ahead and talk about this. So, well, wait a minute. They're both dominant, but they're different. So this organism is heterozygous. That one's the same, heterozygous, heterozygous, and heterozygous. So our genotypic ratio is 100% HZ, heterozygous. They're different. What's this organism look like? What's our phenotypic ratio? Now in this case, they're both dominant genes. Nobody's going to be able to successfully overpower one another, to successfully win to express their color. Nobody's going to successfully work to do this. So in this case, the red and the purple are going to kind of blend together to form a pink. So if you look over here, they're all 100% the same. So 100% of these birds, instead of being red and purple from this cross, would end up having pink feathers. And this is a concept called incomplete dominance. So if you have the 2 R's, you're red. If you have the 2 P's, you're purple. But if you have one of each, they're both dominant genes. They can't, one of them has to overpower one of the other one, but unfortunately they're both dominant. So they can only compromise, and you end up with a blending of pink, and that's how that works. The other concept behind this one is a concept called codominance, and this is how you end up with spotted organisms. So let's take a brown cow and a black cow. So a homozygous dominant black cow and a homozygous dominant brown cow, which in this case I'm going to give R because 2 B's gets very confusing. So a homozygous dominant brown cow crossed with a homozygous dominant brown cow. So let's do our little cross real quick. Homozygous dominant on the top, homozygous dominant on the bottom. And in this case, it's codominant. Neither of these genes are strong enough to overpower one another. Neither of them are strong enough to be expressed fully, so they just compromise. I'll take half the body, you take the other half. And it just kind of happens to depend on wherever the sperm and eggs end up after the crossing over processes and during the building of the body. So anyway, let's cross these. Big B. Now in this case, instead of being a brown and black cow that's kind of halfway chocolate colored, you end up with a spotted cow that has black spots and brown spots intermixed with itself. And this is the concept of a roan horse, and this is how their color works. They have hair colors for both red and white, or gray and brown and black and white and things like that at the same time. So they express both of the colors equally at the same time, just in different areas. So it's very interesting in codominance. So I'll tell you if you have a codominant trait or an incomplete dominant trait when it comes to your exams. So, so another concept is pleiotropy. I'm not going to worry too much about pleiotropy. It's when one gene impacts more than one trait. So you have things called linked genes that if they don't function properly, you're not going to be able to produce certain proteins that you need to be able to form a functioning body. So your body might look slightly different, or you might have a genetic pathway defect or something like that. So just because one gene works properly, and it has to impact the other genes down the stream of it. So if it's not working properly, you might have defects in other genes, even though those genes might not technically be broken. So it's a very interesting concept of pleiotropy, where more than one gene impacts more than one trait. So let's go ahead and finish this up real quick on the concept of sex-linked traits. This is a very interesting concept. When you have an offspring, you have male's offspring and female offspring. So a female offspring has 2 X's, a male has an X and a Y. This is for their sex chromosomes, by the way. There are genes that are carried on that sex chromosome. The gene for lots of things are carried on the X chromosome, so the X chromosome is very useful. Lots of good info on the X chromosome. On the Y chromosome, however, there's only one really important gene, and that gene makes you a male. It gives you testes. It's called TDF, testes determining factor. It's the only gene that's on the Y chromosome that matters. So that's about all it's good for. Anyway, so the traits that are carried on the X chromosome are really important. So if you're a female, you have 2 X chromosomes, which means you have a trait carried on that X chromosome, which is abbreviated by a little risen up letter up here, and the other traits are represented like that. And same over here. So X's are given the little uppercase there, and Y, well, Y doesn't have any genes on it. There are no genes on the Y chromosome other than that TDF. So males only have 1 copy of the genes that are carried on the X chromosome. So Y doesn't get anything. So we can talk about diseases that are carried in sex-linked genders and things like this. So things like male pattern baldness, hemophilia, and things like this. So that are more prevalent in males than they are in females. And let's talk about why. It's a very odd concept. Actually, so let's talk about hemophilia. So hemophilia is a gene, a recessive gene, a disease, I should say, that impacts the clotting of the blood. Hemophilia is a recessive gene. It's also a recessive disease, which means if you want to have, or if not want to, if you have hemophilia, you're going to have 2 recessive copies of that gene in your genome, which gives you hemophilia. If you have hemophilia, if you get cut, your blood will not properly clot itself, and you can bleed to death very easily. So if you have anything other than a double heterozygous or double homozygous recessive like that, if you have 1 big H or one H and little h like that, you will not have hemophilia. You have to have 2 of the little h's to have hemophilia, and that's how that works. Well, unfortunately, the gene for hemophilia is carried on the X chromosome, which means it's marked up here. So a female that is homozygous dominant for hemophilia would be designated like this. Homozygous dominant, a female that's homozygous dominant. Well, for one, we know it's a female, so we're going to have to write 2 X's down because of that. So if I tell you gender, you know we're talking about a sex-linked trait, which is when you know you're going to have to bring out the X's and the Y's and do these little caret letters and work on the Punnett squares this way. So a homozygous dominant female for hemophilia. So let's go ahead and add in what that stands for. Homozygous dominant. Well, homozygous dominant is 2 of the big letters. So 2 of the big letters for this trait are H's. So 2 big H's, 1 on each X chromosome. This female does not have hemophilia. If she did, she'd have 2 little H's. She does not. She also cannot donate a little H to her offspring, to her eggs. She can only donate a non-hemophilia carrying egg. So that's a really handy thing. Now in males, on the other hand, unfortunately how it works is this. Males have 1 X chromosome and a Y. So in females, if you have a defective H, a lowercase h, if you end up with a recessive gene for hemophilia, it's ignored because you have 1 dominant gene in your other X chromosome to overpower it, to give you a fully functioning version of the gene to give you your blood clotting ability. Well, if you're a male and you end up with 1 of those defective copies, that's all you got. You have no other H over here on your Y chromosome. There is no other gene at all to counteract that H. So males are significantly more likely to get diseases like hemophilia from the process that I'm about to show you due to the fact that they only have 1 X chromosome. So let's go ahead and cross this. So let's take a female. So we know we got a female that is a carrier for the disease hemophilia. So in this case, I haven't introduced the term carrier yet. Carrier is the same word for heterozygous in sex-linked traits. It's the exact same word. So heterozygous for hemophilia is essentially what I just said here. A carrier for hemophilia. She's heterozygous, which means on one of her X, she has the big H, and the other one is a little h. She carries the gene for hemophilia. However, she does not have hemophilia herself. And let's cross her with a normal male. And that's the term given to males that do not have the trait in question, hemophilia, male pattern baldness, or whatever. They're normal. Males cannot be carriers because they either have it and they're affected, or they do not and they're normal. So males cannot be carriers. So let's cross this carrier female for hemophilia with a normal male, which is X with a big H for non-hemophilia and a Y with no prefix there. So let's draw our monohybrid cross and figure out what the chances of these two people having a child with hemophilia are. Okay, so let's go ahead and break this up. X and the big H goes with it in our first letter, so big X and H. So X and H goes over here with a little h in it this time from the other X. Down here we put the male, a big X, and a big H for hemophilia. And in this case, a big Y with no other letter on it. Let's do our cross. Big X, big X, big H, big H, big X, big X, big H, little h. Big X, big H, big Y, big X, little h, big Y. Okay, genotypic and phenotypic ratios. Let's go ahead and talk through these. These are super easy. How many carriers for hemophilia do we have? How many males, how many females? So our genotypic ratio is, we got 1 female, 2 females, 1 female normal, 1 female carrier. So 1 NF, 1 normal female, 1 carrier female, 2, oh, here's a male now. So X and Y, it's a male and he's normal. To 1 normal male, to, oh, this poor guy, he has hemophilia. To 1 male impacted or affected. Or you can just put H for hemophiliac. So we know this poor guy down here, he doesn't have another X chromosome here to counteract his defective little h from hemophilia. So he's stuck with hemophilia. He does not have a way to get rid of that recessive gene. He doesn't have a dominant gene that can overpower it. So he's just stuck with whatever the X that he has is, and that's how sex-linked traits work. Male pattern baldness works this way. So the only way you can have a female with hemophilia is if you have a male that is a hemophiliac mating with a female that is a carrier for hemophilia. So XX with a little h. So let's do that cross real quick. Big X, H with a little h for our hemophiliac male. Y, big X with our big H, and big X with our little h. So let's do our cross here. Big X, big Y, H, little h, big X, big H, Y, big X, little h, big Y, H, X, Y, Y, H. Okay, so that's done. So in this case, we have a female that's a carrier, a male that's normal, a female that's a hemophiliac from both. She has to have 2 copies. Remember, it's recessive. You have to have both of them to be recessive. If she has one dominant, it overrides it. And then 1 male that doesn't have, that has hemophilia, doesn't have anything on that Y. So that's the only way you can have a female with a sex-linked trait, is if the male happens to have this disease itself. Now, hemophilia is a big deal. A long time ago, males used to not live very long. You're more than likely to bleed to death in childhood if you had hemophilia. But traits such as male pattern baldness, color blindness are more prevalent in males. They are carried on the X chromosome. Females do end up colorblind from time to time. With a female pattern baldness, because those traits don't really impact your ability to survive and successfully pass on your genetic material. So those carrier females and things are pretty common. So every once in a while they'll mate with a male who is impacted with male pattern baldness or color blindness, and end up with females that are colorblind, or also that have color pattern baldness, which is an interesting little concept. So that's sex-linked traits in a nutshell. I think the last little bit we got is the concept of X inactivation, which is an interesting concept. And this is how cats work. So if you're a cat, you got your chromosome here, and you got an X chromosome from your mom and an X chromosome from your dad if you're a female. If you're a male, you got X and Y. Now the gene for cats, for hair color on cats, is colored on the X chromosome. So you can have 4 different color combinations on the same in the same cats. You can have black, brown, orange, and yellow. In males, well, it's carried on the X chromosome. You can have brown and black, or black and brown. And the Y can't carry any colors. Y doesn't carry any genes, unfortunately. So, or anything other than testes determining factor. So there's only 2 color choices to choose from in males. Now, this is why female cats are all calico. When this cell divides and produces a sperm cell, the X, or the, whatever you call it, the eggs and sperm are going to get different, or the egg cells are going to get 1 copy of this, and the cell can turn on or off the brown or orange randomly. You don't need 2 copies of the same color gene, so the cell is going to turn off one or the other. So this particular set of cells over here might become brown. It turns off the black cells. The cells right next to it might turn off the brown cells and become black. The cells over here might turn off their orange cells and become yellow, whereas the cells right next to them might turn off their yellow cells and become orange. And that's how female cats can have multiple colors inside of them at the same time, whereas male cats, they can't do that. Male cats tend to be locked into 1 or 2 colors and very slight variations on that same color. So females, with that concept of X inactivation, turning off one of their X chromosomes because they don't need it, allows them to have more colors inside of them than they normally would. Now this concept occurs in every female of every single species and every single cell, because you don't need 2 functional copies of hair color genes. You just don't. You'd have twice as much hair than you needed, or twice as much color pigment as you needed. So they just turn off one of the X chromosomes. Now males obviously don't have 2 X's, so we just use the one that we've got, which is what influences that sex-linked trait as well. So you can use a pedigree to look at relationships in families. Very easy to look at this. Very, very, very simple to understand as well. Female, male. Normal, sick, or affected. Carrier. That's how you read a pedigree chart. So what you do, you can read a pedigree chart by looking at the mother and the father. So mother and father, you breed them together, they produce offspring. Okay, so we know that, they're going to have 4 kids, and we're going to make everybody but the male normal. So what we're going to do here, you can see that the male is affected, and the male here is also affected. So let's have these guys have a baby. Let's add in another square here. And now let's have these guys have babies. So let's have 2 kids for them, a male and a female. Oh, and the male is sick in this one as well. Male is affected. Well, in this case, how do we know what's going on here? Well, if this male was able to get this male sick, but these females aren't. It's obviously not a dominant gene. Something's going on here. This male's sick, but his dad's not, neither is his mother. So why is he sick? Interesting here. This is where you can read, if you know how to read a Punnett Square, you can fill in what's going on here. What's going on here is this is a sex-linked trait. This guy's mom is a carrier. That's how that works. So she donated her X chromosome. That was defective. And even though his dad didn't have it, he gave a Y chromosome that was unable to counteract that X. That gave it a defective X. Which also means that since the same thing happened here, the male is sick, the mother doesn't even have to be a carrier. She donated that defective Y. So she has to be a carrier as well. She has to be a carrier because we know that her female carrier or female daughter also carries that gene. So this is a very interesting little way to read a pedigree chart. Not that super useful for anything other than studying genetics. The environment can influence genes as well. You can probably figure this one out based on things like your skin color. When you go out in the sun and you get suntan, now that's more influenced by DNA damage. But the environment can influence how much protein you produce after you've damaged your DNA to repair it. The temperature that eggs are incubated can also change the gender of certain organisms like lizards and reptiles and things like that, can alternate or alter the genotypic and phenotypic ratios, which is interesting. Okay, so that's pretty much all I've really got for you guys over this lecture on patterns of inheritance. I hope it's useful. If you guys have any questions at all over this, feel free to send me an email. If not, have a good rest of the day.